Okay, this is where we left off with this concept of hypotonic, hypertonic. And then with a hypotonic solution, what's going to happen is the cell is going to rupture. And we particularly think of this when we think of red blood cells. And so it's called hemolysis. The red blood cell will rupture. And if that happens, or crenate, and if that happens, to any degree, then that potentially can cause anemia, that can cause death with regard to that. So the tonicity or the osmolarity is important, and this has to do with not only the water compartments but the amount of what is dissolved in there, so the solutes. Both have to be in homeostasis for the body to be healthy. And then you have it again here. Again, this is from the book. Maybe something that you need to look at a couple of times and a little bit to have that in your head in terms of hypotonic, hypertonic solutions. And you do a little bit of this. You do this in this is what you're doing in the egg lab if you're doing the egg lab in your lab class. Then a couple of other transports. You have endocytosis, where you're taking those ligands that are in the cell membrane, so the plasma membrane, and actually taking in you can take in particles or compounds that are right outside of the cell membrane. So if it's right outside of the cell membrane can actually close around it and bring it in, endocytosis. This is going to be receptor mediated, meaning those ligand receptors are going to be sensitive to having that particular compound right outside. This can be cell drinking or cell eating. So endocytosis can be two different types: cell drinking, so taking in fluids, or taking in solids, so pinocytosis or phagocytosis. And this would be considered vesicular or vesicular transport. And here you have it explained. This is with regard to the book. You have your ligands right there. And those are going to be unique or specific. You have specific compounds reacting with those receptors, and as they become full, it causes that membrane to fold in on itself. And in folding in on itself, it breaks off from the cell membrane itself. So the cell membrane folds in, pinches off, and comes on in. That can happen in the opposite way too. So here you have you have pinocytosis and phagocytosis. It can happen the opposite way too, that you have exocytosis too, where you have a vesicle. This is going to come from the Golgi apparatus. That vesicle, it comes close to, butts up against the inside portion of the cell membrane, becomes one with the cell membrane, and then the contents are released from the cell, exocytosis. And this can be solids and also liquids, but mostly solids. This transmembrane potential is another concept. I just had space on there and I wanted to use the space. What happens is you have sodium, potassium, you have these charges. We talked about the sodium-potassium pump. Well, if you have far more sodium outside the cell than inside the cell, you do have a charge to that membrane. So you have more positive charge outside than inside, because sodium, potassium have different charges. And what happens is that transmembrane potential are the unequal charge across the plasma membrane, but that unequal charge can be a resting charge. So you have a negative 10 millivolts to a negative 100 millivolts depending on the cell type. And in order for something to actually move across, particularly when we think about an electrical impulse in the nervous system, that transmembrane potential shifts because you have a shifting of the ions between the outside and the inside of the cell membrane, and that then creates what's known as an electrical impulse, and we do talk about that in the nervous system. But it's important that you start getting these concepts in your head, this transmembrane potential, that the membrane, it does have a charge, but that charge can be the resting charge. All right, the last thing to talk about is mitosis. Mitosis is soma cell replication. Most of the time the cell is just being itself, but replication happens in 3 stages. And so prior to interphase, or interphase basically is a non-dividing, first thing that happens is DNA replicates itself. Second thing is mitosis, and then you have the cytoplasm. So all of the things that are in the cytoplasm replicating and dividing into 2 daughter cells. So we call this cytokinesis. So first part, DNA replicates. Second part, you have the DNA going to opposite poles. Third part, all of those organelles that we just talked about replicate, and the cytoplasm splits. DNA unwinds. You make a duplicate copy of it. You have 2 exact strands of the same DNA. That's the first thing that happens. The second thing that happens is you have these spindle fibers and the centrioles. So the centrioles and the centrioles put out these spindle fibers that attach to the centromere of the chromosome and start pulling them apart into separate poles. But before it pulls them apart, so prophase is when the nuclei disappear. Centriole pairs move cells to the poles, so they move to the centrioles move to the opposite poles of the cell, and cause the DNA or chromosomes, the sister chromatids at that time, to line up down the middle. Now, when they are lined up down the middle, we think of that as metaphase. So prophase is the actual disappearing of the nuclear membrane, the nuclear envelope, and then this organization of the chromosomes. Metaphase, the chromosomes are organized, lined up down the middle. And then you have it kind of here again. Again, this comes from your book. And then anaphase, those chromosomes are starting to pull apart. And then telophase, not only are the chromosomes pulling apart, you have that nuclear envelope reforming. You start having a little bit of cleavage, a little bit of remaking of the cell membrane. So that membrane. And then cytokinesis, the third stage. DNA is the first stage. DNA replication, the first stage. The DNA organizing itself and moving to opposite poles of the existing cytoplasm, and then having that cleavage or furrow happening. And then cytokinesis is the third stage of mitosis. Cytokinesis is the duplicating of organelles, so the endoplasmic reticulum, the Golgi apparatus, the mitochondria, everything is going to duplicate, and so that each of those daughter cells have exactly the same thing as each other, and each of the daughter cells has exactly the same thing as the precursor cell, the cell that we started out with. And with that you have mitosis, and that's how every single one of your soma cells replicate. Every single one of your body cells replicate. Then after that replication, or actually a differentiation is kind of a different topic. Differentiation is the specialization of cells. I remember when we first started talking about this, I said I'm talking about generic cells. I'm talking about what cells can do. What happens is these all come from your embryonic stem cells. Embryonic stem cells will replicate themselves and then eventually differentiate into heart cells, muscle cells, nerve cells, mucous cells, etc. But all of the cells, whether you're a heart cell, a mucous cell, a gastrointestinal cell, have the same DNA. That replication happens because certain parts of that DNA are active in that cell as opposed to another. After they differentiate, then they can congregate together to form tissue, whether that is liver tissue, fat tissue, neurons. And then with that, by definition, if they've differentiated, certain parts of the genetic code is off or on in that particular cell. And all body cells have 46 chromosomes, except for sex cells, and differentiation depends on this whole process of active or inactive genes in that particular cell. And tissues is what we're talking about in the next chapter, and so this kind of lets us off and picks us up exactly where we should be in order to understand what's going on with the next chapter in tissue.